Hi all, welcome to GFI's 2023 Research Grants Program overview. Through today, we're hoping to be able to give you an overview of our RFP and talk through some of your questions and answers in our Q&A session. So just to go briefly through our agenda, I'll give a brief introduction into our Research Grants Program. I'm then joined by four of my colleagues, which are the topic area leads for the four topic areas that we've highlighted in our RFP this year: Priera Pinescu, Claire Bonkamp, Elliot Schwartz and Adam Lehman. I'll then talk through briefly how to apply and then we'll answer some of your burning questions in our Q&A panel session. Just to give a brief overview into GFI. GFI is a 501c3 nonprofit developing the roadmap for a sustainable and secure and just protein supply. Powered by philanthropy, we focus on three key areas of research. Our science and technology team, who are the team that are joining you today, advance foundational open access research into alternative proteins and create a thriving research and training ecosystem. We also have a corporate engagement team which partners with companies and investors across the globe to drive investment and accelerate innovation and scale the supply chain. And finally, our policy team advocate for fair policy and public research funding into alternative proteins. GFI has earned the GuideStar's Platinum Seal of Transparency in 2019, 20, 21 and 22. This is obtained by less than 1% of nonprofits and it reflects our commitment to maximum impact and efficiency and inclusion. Across our 6 affiliate regions, we employ over 190 staff and we work as a force multiplier, giving our expertise to departments around the world. Now, to go into GFI's Research Grants Program, so GFI works to accelerate the development of alternative proteins and help make products that are delicious, affordable and accessible. This is our theory of change. GFI defines alternative proteins as those that are derived from cultivated meat, plant-based products and fermentation, and these all offer exciting research opportunities and have a tremendous positive impact on climate change and global health. Through our research funding program, we aim to provide dedicated funding to address the scientific challenges associated with our theory of change. We aim to build a community of alternative protein researchers, and we aim that this research funding can act as a springboard to unlock further funding from elsewhere. To date, our program has supported over $17 million worth of awards, and we have over 100 research projects across 17 countries. This year's RFP highlights 4 topic areas, and we'll be going into each of those in more detail through this webinar. This year, we're offering up to $3.75 million across these areas, and the call will close on September 21st later this year. The topic areas we've highlighted work across all 3 production platforms that GFI supports, and awards will be for up to 24 months in duration and $250. As GFI aims to promote and support the research funding ecosystem and the researchers ecosystem, we aim to support collaboration and bring new people into the field. And for this reason, we are offering up to an additional $100 per award for projects that involve collaborations with researchers that are new to the field. And with that, I'll hand over to Pri, who will talk through our first funding priority, Extrusion 2.0.
Excellent. Thank you, Sam, for that intro. I'm excited to tell you guys about our first priority topic focused on Extrusion 2.0, enhancing traditional extrusion through process innovation and mechanistic evaluations of protein texturization. So I really hope that this audience, whoever is interested in this topic, is already pretty familiar with extrusion. But just as a way of introduction, we know extrusion is the most popular texturization method for plant-based meat production, whether this be high moisture or low moisture extrusion, where low moisture tends to be used a lot more than high moisture, but both are really valuable in the field. Right now, extrusion does have a current output of up to 500 kilograms per hour, which is much better than a lot of other texturization techniques that could be used for plant-based meat, but could definitely be improved. And additionally, the current capabilities are being able to form protein extrudates with, on the order of dozens to about 100 micrometer fibers. Within the extruder, there's definitely a few things that can be done to optimize either the texturization portion or the high throughput portion. 2 sections that can definitely be called out are the screw section, where there might be a single barrel or double screw extruder. And this can change the properties of the protein ingredients that are fed through. Additionally, die sections help control the shape and solidify the product. And then, of course, there's other things that affect the product, like hydration time, order of addition of ingredients, etc. But these are kind of the main highlights. And in this grant, our goal is to improve extrusion by improving its throughput capacity, as well as provide innovative strategies to replicate texture and visual appearance of conventional animal muscle cuts. So diving a little bit further into each of these, next slide, please, Renee. Thank you. Let's focus on the texturization portion, that 100 micrometers, maybe a few dozen micrometer fiber length scale. We know that extrusion is a top-down texturization method, so there are some bottom-up texturization methods like spinning that could be used as well. But the reason why extrusion ends up being so scalable is because it is that top-down technique. So how can we make these top-down techniques even better at replicating animal muscle cut structures? Additionally, because extrusion has a lot of parameters that are coupled to one another, it's very difficult to elucidate how proteins interact with themselves and other ingredients under various temperature, moisture, and mechanical shear conditions. There are some really great current efforts, and these are really good examples of the types of projects we're looking for here, where researchers are creating software that's coupled with unique dye processing. They're optimizing dye geometries and even applying rotating dyes to be able to enhance the structure of an extruded plant protein. So that's exactly what we're looking for. It's how do we get that top-down extrusion texturization to be even more refined to produce geometries that could be even more modular. Next slide, please. And part of what drove this RFP or this priority here is the fact that we have run a few studies at GFI where we really emphasize that scaling plant-based meat production with current extrusion capacities is going to require massive infrastructure investments. So we have two analyses linked here. I highly suggest taking a look through them just to see the motivation for this topic. So for example, here on this graph, I'm showing one of the analyses where capturing 6% of the meat market would require an estimated 27 billion CapEx, not even the operating expenses, just the capital expenditure, to build at least 800 facilities with roughly 2,000 commercial scale extrusion lines. This is obviously a lot of capital that would be needed in order to build this infrastructure. This is just a lot of infrastructure, 800 facilities, 2,000 extrusion lines, right? So being able to take this and say, okay, what else can we do besides just putting more steel in the ground, more steel equipment in our factories? What else can we do to optimize this process? These studies do not take advances in extrusion into account. So they assume that the capacity that we have now would be the capacity that we would have in 2030. So I really think, we can open our minds and think, oh, wow, how can we increase the throughput even more to maybe lower this $27 billion estimate? Really recently, it's not in our written RFP, so I included it in this presentation as a little Easter egg for you guys, but Clextrol just released a document that showcases a high capacity dye that actually has a capacity of up to 1,200 kilograms per hour. So if you remember the first slide, I said the capacity was up to 500 kilograms. So we're already seeing these advancements in place. And again, I highly recommend looking at that to see an example of something that would be really fantastic for us to look at in this priority area. Next slide, please. So really, what are we looking for in this proposal? Please let us know how your approach will help improve extruders, meet organic, help plant-based meat organic organoleptic parity with conventional animal products, and how will it make extrusion more high throughput? So successful proposals will articulate this, and proposals that focus on traditional methods to analyze plant proteins or other ingredients will definitely not be accepted without this explanation. So please really show how it is an extrusion 2.0, that extra level above what we already see. Please say what plant proteins and ingredients will be evaluated, how the improved organoleptic properties or scale will be evaluated at the end, and then describe the biggest challenge. Be realistic. We all know as researchers that things go wrong. So what could go wrong and what are your mitigation plans for that? Additionally, we love proposals that really lay out the financial and environmental costs for the research that's being done. So innovations at pilot scale or ones that include life cycle or techno-economic analyses in general tend to really strengthen proposals. And with that, I will pass it on. Thank you so much.
Thanks so much, Pri. I'm excited to tell everybody about our second priority, which is developing tools and knowledge to promote stemness and proliferation in seafood cell cultures. Next slide, please. So just by way of some background as to where we are now with fish and aquatic invertebrate cell cultures. So this says fish on the slide, but very much applies to invertebrates as well. There's a lot of sort of challenges when you go into starting a fish cell culture experiment. So everything from long doubling times to issues with spontaneous differentiation, issues with media, both in terms of the cost and just the lack of appropriate, especially serum-free culture conditions, lack of appropriate cell lines even to start with, and low or sort of not well understood metabolic efficiency. And then on the flip side, where we want to be on the next slide is kind of the opposite of everything I just said. So we'd like to have cell lines with, or cell line and media combinations with relatively short doubling times, easy to access off the shelf cell lines, kind of all of the things that are, I don't need to read through all of these, the opposite of everything that I just said. And so on the next slide, what we're really trying to do with this funding priority is to build as much of this kind of metaphorical bridge between this current state and the future state that we want as we can. And so this really means bringing together ideas, people, and funding to address these challenges. So next slide, please. And what this means more concretely for this funding priority, people, of course, being all of you, the researchers who are best positioned to address these challenges, funding from GFI donors, who we very much appreciate, as always. And then on the ideas front, I think there's a lot of kind of different and complementary approaches that one could take to solving these challenges. And this is by no means an exhaustive list. So, you know, everything from traditional media optimization just through screening. We could think about, you know, things like transcriptomic studies to better understand both kind of the fine distinctions between cell types that exist in fish and aquatic invertebrates, which I don't think we do understand completely. And also kind of some of the markers that are happening as cells are differentiating or proliferating, and kind of really getting into the nitty-gritty of kind of what's going on in these cells, to kind of go beyond just, you know, looking at a few markers, looking at, you know, morphology under the microscope, things like that, optimization and development of cell lines, and then kind of underlying almost all of these approaches is the need for better optimized research tools. So things like antibodies for identifying cell types, annotated genome sequences for a variety of species, all of those things are kind of going to be necessary for, depending on the approach, you know, to be able to even do those experiments. Next slide, please. So in terms of what we're looking for in proposals for this priority, the key question is really explain to us how the approach you're proposing is going to make fish or invertebrate cell culture essentially less of a pain. And so how is your proposal going to address the challenges that we talked about? So how will it improve doubling times, metabolic efficiency, improve the availability of high priority cell lines, reducing media cost, preventing spontaneous differentiation during the proliferation phase, and finding good alternatives to serum and other animal-derived media components? And how are you going to kind of hopefully address a few of these at the same time without unwanted trade-offs in others? And so in terms of the exact approach you take, that's really up to you. And so we really want to cast a wide net as to what are going to be good approaches here. And we kind of need everyone's brain here. We don't want to dictate from GFI's perspective. This is the approach because y'all are the experts. And so another thing that we really want to focus on in this RFP or in this priority is what research tools are necessary for your proposed approach to be successful. Do those tools already exist? Are they accessible? And if you need to create new tools, which we very much encourage, what's your plan for creating them? Is it feasible? And how do you plan on sharing them with the rest of the research community? In terms of species focus, we're looking for proposals that are primarily focused on either fish or aquatic invertebrates. Of course, prioritization of species is going to depend on a lot of things in terms of, compatibility with a food product, potential environmental impact. So if you want to kind of give a little bit of justification in your proposal as to why you chose the species you chose. And of course, availability of cell lines is certainly one factor, but kind of balancing the different considerations when it comes to species. And we will consider approaches as well that take a comparative approach. So if you're characterizing some aspect of your fish cell line and you want to do a comparison with C2C12 cells or bovine satellite cells, something like that, to really kind of understand, because we understand mammalian and to some extent avian cells better than we do fish or invertebrates, can we port some of that knowledge over? We definitely will consider proposals like that, but we're looking for ideas where the primary focus is really on seafood. Next slide, please. So a couple of places you can go to kind of dive into some reading related to this priority. So our solutions database has an entry on promoting stemness and proliferation in fish cell cultures, which is, if you read that in the RFP, it's very similar. The RFP is basically this expanded to include invertebrates. So if you've read the RFP, you've pretty much read the solution. And then we also kind of expanded things to have a little bit more of a focus on tool development. And that focus actually comes from a workshop we did back in January, which some of you, if you attended, you might recognize some of these slides from today. And one of the really, really clear learnings that we took away from that workshop was that tool development is a bigger obstacle than we realized. And so that's why there's so much of an explicit focus on that in this priority topic. And so if you want a little bit more background of the workshop summary, we'll put these links, I assume, in the video description or something as well. And I can drop them in the chat now. And then there's a couple other entries in the solutions database as well that can be somewhat relevant. So we have one on research toolkits, and this really gets to kind of some of the research tool development aspects that I mentioned. And then there's also an entry on species-specific genomic studies. So depending on your approach, those could be worth reading as well. And with that, I believe that's the end of my slides. So I'll pass it over to Elliot to talk about our third priority.
All right, thank you, Claire. My name is Elliot Swartz. I'm one of the scientists that work on cultivated meat at GFI. And for this third priority area, it's about data collection and curation to inform the development of genome-scale metabolic models for the optimization of feedstock formulation and feed conversion. And so I'll just say that, for the slides that I'm providing additional context to, they're all sort of in the context of cultivated meat. But really this priority and the goals and objectives apply equally to fermentation as well. And so we're really looking for proposals here that can enhance cultivated meat development as well as fermentation. Next slide. So with respect to cultivated meat, one of the biggest challenges is the cost of the cell culture media. And lowering the cost of the cell culture media is really crucial to ensuring a future cost competitive cultivated meat industry. And there are really two large buckets of research and development that need to happen in order to reach this sort of future goal. Now, the first bucket is really focused on just simply sourcing lower cost ingredients that go into the cell culture media. And this R&D is very much underway throughout the industry today. And this is really focusing on optimizing for a dollars per liter metric. And so thinking about how we can source lower cost ingredients from switching from things like farmer grade ingredients to food grade ingredients. Now, the good thing about this first bucket is that it has cross-industry relevance because everyone, researching cultivated meat will ultimately use the lowest cost amino acids and other basal media components for their research and development purposes. But the second bucket, which is focused on using fewer ingredients, I think is a harder and longer challenge to solve for the industry. And this requires a switch from optimizing around this metric for dollars per liter to optimizing for a feed conversion ratio, or the grams of ingredients that are required to output 1 kilogram of meat. And this is more challenging because each of this needs to be tailored to each company's cells, cell lines, or ultimate end products. But really, success in both of these areas are required. If you just hit the next animation, Renee. So essentially, what we need to achieve in this future state is efficient media use that's tailored to individual cells or organisms' metabolic needs. And that's really going to be crucial for lowering the cost of both cultivated meat and fermentation-derived foods. Next slide. So just to provide a little bit more, concrete example of the importance of feed conversion ratio with respect to cultivated meat, we can refer to this study that we published earlier this year, which is a life cycle assessment of future cultivated meat production. And in this study, we formulated 3 different media use scenarios based on data that was collected from multiple companies in the industry. And so this baseline scenario here highlighted in red sort of serves as an important and useful benchmark for understanding the quantity of amino acids, sugars, and other media components that we could expect for outputting 1 kilogram of cultivated meat. Next slide. And essentially what we found in this research was that around 600 to 800 grams of solids in the media would be required to produce 1 kilogram of cultivated meat. This assumes that about 70% of meat is water. But the challenge is that this really needs further experimental validation. Now, the good news is that achieving this sort of baseline scenario in the future would result in a feed conversion ratio that's nearly 3 times as efficient as chicken production. So this serves as a really useful benchmark for R&D to move toward as we, as the industry develops. Another important aspect of the feed conversion ratio is its influence over the sustainability of the production process. And so if you compare the high medium scenario here to the baseline scenario in this study, the carbon footprint would increase around 73 to 240%, depending on where and how renewable energy is used in the supply chain. So this is to say that the feed conversion ratio and optimization of the feed conversion ratio is absolutely crucial to ensuring a lower carbon footprint and ultimately lower sustainability metrics for the production of cultivated meat, as well as fermentation-derived foods. So on the next slide, we'll talk about how we think we can really do this, which is by leveraging metabolic modeling to optimize cellular nutrition. You can click through this slide, Renee, thanks. So essentially, this is outlined really nicely in this review paper that I've cited at the bottom that was published by a GFI research fellow, Sandra Romero, just a few weeks ago. And this figure is from this paper. And essentially, the challenge today is that the development of these feedstock formulations is really long and challenging because they contain many ingredients and they have to be tailored, as mentioned, to the certain cell lines, species, or organism that you're working with. And so fermentation really has had a head start where, many different studies and organisms have been studied to create metabolic models to optimize feedstocks. But this is much further behind when it comes to mammalian cell types. And generally speaking, what we need to do is first have a complete data set upstream in numbers 1 through 3 here that allow us to access the annotated genome and metabolic pathways of different organisms, to understand the biomass composition of the cells or the organisms that we're working with, as well as have access to omics data that allow us to sort of plug in to understanding and creating a model for developing the composition of the media, as well as optimizing that media further for its downstream use case, such as biomass accumulation or yield. And so essentially what we're looking for in this study in this priority area are proposals that accumulate and curate the data shown in numbers 1 through 3, allow for the creation of a genome scale metabolic model that can then be leveraged by others in the field, as well as ideally have additional experimental validation through steps like through experimentation, such as it's shown in steps 5 through 7 here, that allow you to iterate and refine this model to eventually, develop a composition or a feedstock or media that's going to be lower cost or more efficient and matched to the metabolic needs of those cells. So I definitely encourage folks to read through the information contained in the RFP, which goes into greater detail about some of the needs and directions here that we're thinking of. And with that, I will pass it to Adam to discuss the 4th priority area.
Thanks, Elliot. Thanks. I'm Adam Lehman. I'm one of our fermentation scientists here at GFI. And I want to tell you about our funding priority D, which is rather similar but distinct to the funding priority C, which is to improve feedstock availability for food fermentation for both precision and biomass platforms. You can move to the next slide. So fermentation as a platform, it typically uses in commercial settings highly refined sources of carbon and nitrogen to feed these microbes and ultimately make the food products or other bioproducts that we enjoy from biomanufacturing. And much of the carbon that we use comes from glucose, and glucose is refined whether from wheat or sugar beets or sugar cane or corn maize. And this refining process is time consuming, expensive, has high global warming potential, but also takes away from opportunities to use these ag streams for human food consumption. Similar but different in using nitrogens that has been sourced from something like a Haber-Bosch process, we are using lots and lots of global warming potential and carbon output to produce highly refined ammonia using mostly natural gas and methane. So we can use microbes in a variety of different ways. Microbes are relatively permissive in their carbon and nitrogen sources. For example, you can feed these starchy residues or potentially carbon gases, or even nitrogen sources in the form of something besides ammonia, something like amino acids or proteins. And microbes have enzymes and metabolic repertoires that allow them to break these down and then use them for fuel to build biomass, or ultimately to create the precision fermentation products that we need. So the idea that we could use lots of non-conventional feedstocks for fermentation is not a new one. However, in food, we don't necessarily always enjoy the same advantages that other bioeconomy platforms do. So for example, if you're using a distillation or refining process to take a biochemical away from a fermentation, you can usually leave some of these side reaction production and things like that behind. And if we move to the next slide, we can see that in food, we don't enjoy that luxury. And one of the big issues that happens is that when we combine non-canonical side streams or feedstock streams with microbial fermentation, is that we often introduce the opportunity for toxins or chemicals or other unwanted molecules to get into the food system. And so I can lead you through a few examples here to think about, and these are the types of solutions we'd like to have solutions to reduce these types of food safety challenges. So often, if we're thinking about using something like an ag side stream, some of those ag side streams are treated or stored in a manner that allows other microbes to grow on them during that time. When that happens, so to take the example of something like peanuts or corn on the left, we can see that some of the microbes that grow on these introduce things like aflatoxin. And so aflatoxin is a molecule that's made by fungi that grow on these during storage and then can bioaccumulate within that feedstock and then pass through into the food system and go into the human and have effects like liver damage and toxicity there. This can happen in a variety of different pathways with bacteria toxins, and this can also happen with the bioconversion of particular feedstock components when you have a fermentation organism on them. So if you're thinking about an example of something with lignin, you could have furfural or furfural-containing foods that get entered into the system, and this can also be a toxin and a bioaccumulator in humans that also ultimately leads to toxicity. And so in food, we have to avoid situations that bring toxins either from the feedstock or toxins that are created by the fermentation organism into our food system while we're trying to valorize some of these novel foods, novel feedstocks. So, next slide, please. So what we're looking for are new fermentation microbes, bioprocess, or feedstocks that avoids some of these issues. So just to kind of summarize through some of the high level things that we're looking for is we're looking for microbes that can metabolize sugars and polysaccharides or starches that aren't just glucose. So we'd like to move to a food system that's more sustainable, and we'd like to be able to do that by using non-canonical carbon chains. Now, many people think about breaking down starchy residues, but obviously within this and in scope here are microbes that can grow on things like waste alcohols, acetate, methane, carbon monoxide, or carbon dioxide, and use these carbons to build up feedstocks, either for biomass or precision APs in a novel way. Similarly, I think feedstock and microbe are very closely tied here. So we're looking for feedstocks that match particular microbes that are able to bioprocess these and bioaccumulate and grow on them in sustainable, but ultimately also in very safe ways. And then finally, part of this can be the bioprocess that matches the microbe with the feedstock and allows them to grow. Or it could potentially be a bioprocess approach that pairs these in a way and then may introduce a toxin that gets safely and highly effectively eliminated so that you could create a product that ultimately was able to pass a generally regarded as safe barrier. Next slide, please. So ultimately, what are we looking for? We're looking for proposals that articulate what fermentation microbes and feedstocks will be tested, characterized, and developed, and what evidence there is for this match of microbe and feedstock to create a safe food product. So the potential for the food safe fermentation process has to be recognized and evaluated during this proposal. We're also looking for price sustainability and availability advantages of the feedstock over the current state of the art. It can be well and good to use a novel feedstock. However, if that feedstock has costs that aren't advantageous over the current state of the art, or if that's a feedstock that may not be available in any commercial quantities that allow it to be scaled, this would be a tough ask to build a food component around it. We're looking for either microbial, biochemical, or molecular evidence of metabolic compatibility between the microbe and the feedstock. And then also a clear-eyed technical and biological association of the challenges with that particular bioprocess. So really what we would like here in this is to see something that works particularly well for food and allows us to really diversify out the feedstocks that we're using. There are going to be multiple solutions within the world of feedstock and microbe, and we look forward to exploring them within this RFP topic. And I think with that, I am passing everything back here to Sam.
Thanks so much, Adam. So if all of those 4 areas, if any of those areas seem of interest to you and you're interested to learn about what the next steps are and how to apply, I'll just talk briefly through that part of the process. Next slide, please, Renee. So in order to apply for our research grants programme, you need to submit your proposal through our application portal, which is at grants.gfi.org. In order to access the application form, you'll need to set up a user profile. And when you get to the application form, there'll be a number of documents that you need to submit. The initial form that you'll need to fill out will include just standard project details, which include your project title, summary, and abstract. The summary and abstract in there, you should be typing those with a view to an external audience seeing them. So for successful proposals that we fund through our RFP, we'll be using that information as part of our external communications, and they're just short summaries of what your project is. Your full proposal will be included within the remaining documents. The case for support will be up to 6 pages, which includes up to 1 page to go over the track record of the applicants and the partners within there. So 6 pages, 5 pages for your case, and then 1 page for your track record. There'll also be a one-page work plan outlining your plan for the proposed work over the duration of the project. A justification of resources to justify the resources that you've requested through this proposal. An impact plan, which is up to 2 pages describing the proposed impact and your plan for that to occur. And there's also an area for your project partners' letters of support. So these are partners coming in with you to the project that will be making contributions either financially or in kind to the project. These are not required for the RFP, but you can certainly upload them if you do have partners joining with you. Proposals will be assessed using our standard assessment criteria. So we'll be looking at the scientific excellence, the applicant and partnerships, the expected impact, and the project planning. We recognise that not all proposals will be written by native English speakers, and we will certainly take that into consideration when we're going through the review process. So please don't be concerned if that is the case. So any questions that you may have can be answered in multiple different ways. So please check the FAQ link within the RFP document where we outline detailed answers to some of the most common questions we get about our research grants programme. We're also on hand through our grants mailbox, which is research_grants@gfi.org. And we'll be holding our Q&A panel now, and I'll come back to the process around that in a minute. In addition to this, you can also meet with us one-on-one to ask us questions about your proposal or your proposed proposal. We'll be holding a networking and information session where you can meet other collaborators, referencing our earlier policy on collaborations. But you can also meet with GFI staff. That will be taking place on July 24th, and you can register using the links here, and we'll make sure those links are sent out to you or posted in the chat. Thank you very much. We'll move directly to our Q&A session.